Okay, good morning, students. As we are discussing regarding the morphology of the cell injury, in the previous class we have discussed regarding the different morphological features. Okay, that is hydropic change, hyaline change, and the mucoid change. Okay, today we are going to discuss regarding one of the morphological feature of the cell injury, that is the fatty change. Okay, this fatty change also known as statosis. Okay, this you will also call it as a statosis. This fatty change you will also call it as a statosis. And if you see this fatty change, this you will call it as intracellular accumulation. Okay, it is intracellular accumulation of neutral fat. Okay, see this neutral fat is the triglycerides. If you see, that is mainly the intracellular accumulation of the neutral fat, okay, within the parenchymal cells, where it is storing or accumulation, that is within the parenchymal cells. Clear? See, remember here fatty change. If you see the meaning of this fatty change, that is intracellular accumulation of the neutral fat within the parenchymal cells. Okay, you will call it as a fatty change. Okay, this neutral fat, which is nothing but the triglycerides, or you will also call it as a esters. Okay, three esters of three fatty acids, you will call it as a neutral fat. Clear? See, mainly this is going to be deposited in the cytosol. Mainly all these fats are going to be deposited in the cytosol, and commonly in case of the liver. Clear? Most commonly this neutral fat is going to be stored in the liver. See, why? Because this you will call it as a fatty tissue. Okay, liver is called it as a fatty tissue. So most of this neutral fat is going to be accumulated in the cytoplasm of this liver. Clear? And not only that, this type of neutral fat can also be stored in other non-fatty tissues. Okay, that non-fatty tissues includes the heart, okay, skeletal muscles, and the kidneys. Clear? See, and if you see, I think you have understood what is meant by this fatty change. Okay, fatty change you will also known as statosis. Clear? And this is the intracellular accumulation of the neutral fat within the parenchymal cells. Clear? Here you remember neutral fat means which is the triglycerides or the esters of three fatty acids. You will call it as a neutral fat. Clear? Mainly this will be deposited in the cytoplasm. Clear? Not anywhere inside the organelles, but this will be deposited only in the cytoplasm, mainly in the liver organ. Why? Because this we will call it as a fatty tissue. Clear? And not only that, which can also be deposited in other non-fatty tissues like heart, skeletal muscles, and the kidneys. Clear? See, for your understanding, okay, this fatty change we will discuss with one example of one disease. Okay, that you will call it as a fatty liver. Okay, we'll see in detail regarding this fatty liver. Okay, this is one example we are going to discuss today. We'll see what happens in case of this fatty liver. Clear? See, this fatty liver means here the why only mostly this fat neutral fat is going to be deposited in the liver. Clear? See, why? Because liver is the most common site for accumulation of fat. See, what is the reason behind that fat is most commonly accumulated in the liver? I told you, which is a fatty tissue. See, that means which will be playing a very important role in case of the fat metabolism. See, that's why this liver you will call it as a fatty tissue. Is that clear? See why most of the fat is going to be deposited inside the liver. The reason behind that is the liver is a fatty tissue. See, that means this liver plays a very important role for the fat metabolism. See, that means whatever the fat that will be present inside our body, that is going to be enters inside the liver for its metabolism. Clear? See, that's why most of the fat is going to be deposited inside the liver, so there is a chance of getting this fatty liver. Clear? And mainly if you see, and depends upon the cause, okay, depending upon the cause and depending upon the amount of fat that is going to be deposited, which will be leading to either reversible cell injury or irreversible cell injury, or which will be leading to the cell death. Clear? Depending upon the cause. Okay, depending upon the cause or depending upon the amount of fat that is going to be deposited finally, which will be leading to either reversible cell injury or irreversible, or which will be leading to the cell death. Okay, we'll see what are the causes. Okay, we'll see what are the causes for this fatty liver. Okay, I will tell you what are the different causes. See, mainly this fatty change, okay, in the liver result mainly from two causes. Mainly two causes. The first one is conditions with excess fat. Okay, conditions with excess fat. See, what is meant by this conditions with excess fat? That means exceeding the capacity of a liver to metabolize the fat. See, that means the liver can metabolize the fat to a certain extent. Suppose if that extent is going to be increased, okay, that you will call it as a conditions with excess fat. Clear? See, that means the liver, how much it can metabolize the fat, that only it can metabolize. Suppose if that fat content is going to be increased inside the liver, that you will call it as conditions with excess fat. And next one is liver cell damage. Next one is liver cell damage. See, what is this liver cell damage? That means when fat cannot be metabolized. No doubt fat is going to be enters inside the liver, but because of damage to the liver cells, the liver is unable to metabolize the fat. So there is a chance of deposition of fat inside the liver, which will be leading to the fatty liver. Clear? See, these are the two main causes for this fatty liver, that is conditions with excess fat. That means how much amount of fat that the liver can metabolize, which can metabolize, but if there is an excess amount of fat which is unable to metabolize, that is going to be accumulated inside the liver, which will be leading to the fatty liver. And next one is liver cell damage. As we are discussing, because of damage to the liver, whatever the fat that is going to be enters inside the liver, which is unable to metabolize it, so that fat is going to be deposited inside the liver. Clear? We'll see some examples. Okay, with excess, that is conditions with excess fat. If you see like obesity. Clear? Anybody suffering with obesity and diabetes mellitus, definitely there is a chance of developing this fatty liver. Clear? Conditions with excess fat. If you see the examples, those who are having this obesity and diabetes mellitus, there is a chance of development of this fatty liver. The next one is, we'll see regarding the liver cell damage. Here examples, if you see like alcoholic liver disease, alcoholic liver disease. That means those who are taking the alcohol, there is a chance of damage to the liver cells. Clear? So this also one of the cause for this fatty liver. And next one is starvation. See, because of this starvation also there is a chance of liver cell damages. And next one is hypoxia. If you see hypoxia, which is also one of the condition where the liver cells is undergoing damage. And next one is in case of hepatotoxins. Clear? See, the chemicals are the toxins which can be toxic to the liver cells. The next one is some drug induced. Okay, drug induced damage to the liver cells. Okay, the drugs like, if you see methotrexate. Okay, this methotrexate will be used in the treatment of cancers. The next one is the drugs like tetracyclines. Okay, these tetracyclines are the antibiotic, clear, which can also causes the damage to the liver cells. And next one is protein calorie malnutrition. Okay, protein calorie malnutrition. Okay, these are all the different examples, okay, which will be related to the liver cell damage. Clear? So these are all regarding the causes for this fatty liver. Is it clear? See, I'm discussing what is meant by this a fatty liver and what are the main causes for this fatty liver, that is conditions with excess fat. Clear? The examples like obesity and diabetes mellitus. Clear? Those who are having these two conditions, definitely there is a chance of development of this fatty liver. And next one is liver cell damage. See, these are all the different examples where there is a chance of liver cell damage, ultimately which will be leading to this fatty liver. Next we'll see the pathogenesis of this fatty liver. Okay, if you see regarding the pathogenesis, first we'll see actually what happens inside our body. Clear? Suppose if you see the lipids. Clear? The lipids exist as a free fatty acids. See, this is going to be enters inside a liver. Clear? See how these lipids that is going to be enters inside the liver. Clear? Mainly by two sources. This lipids or the free fatty acids that is going to be enters inside the liver. Clear? The first source is because of diet. Okay, with the help of this diet, the whatever the free fatty acids that are present in the diet, that is going to be enters inside the cell. Clear? How these free fatty acids in the diet which will be enters inside the liver means in the form of chylomicrons. Clear? In the form of chylomicrons and free fatty acids. Okay, in this form, clear, mainly through the diet, the free fatty acids is going to be enters inside the liver. And next one is with the help of adipose tissues. See, that means adipose tissues that will be present inside our body. With the help of this adipose tissue also, this free fatty acids finally enters inside the liver. Clear? Next, what happens? Once inside the liver, we are having the fatty acids. Clear? Through these two sources, finally, the fatty acid has been entered inside the liver. Now, this free fatty acid is going to be converts into the triglycerides. So I already told you fatty change means definitely which will be an accumulation of the neutral fat. Okay, that is important, not the accumulation of free fatty acids. Okay, accumulation of neutral fat means I told you which is a triglycerides or which is an esters of three fatty acids. You will call it as a triglycerides. See, once there will be an accumulation of these triglycerides, then only you will call it as a fatty liver or the fatty change. Clear? See, one thing is this fatty acids, how we are getting, that is through the diet as well as through the adipose tissue. Clear? And next one is as these fatty acids is converted into the triglycerides with the help of alpha glycerophosphate. Okay, with the help of alpha glycerophosphate. Clear? See, and not only that, some amount of acetate. Clear? See, which can also be produce these fatty acids inside the liver. Clear? See, I told you through the diet and through the adipose tissue, the fatty acids is going to be enters inside the liver. But some amount of acetates that will be present inside the liver cells can also produces this fatty acids. Clear? See, finally, this fatty acid will be converted into the triglycerides with the help of alpha glycerophosphate. Clear? See, that means most of the fatty acid is going to be converted into the triglycerides, and small amount, okay, some small amount of these fatty acids will be converted into cholesterol. Okay, small amount will be converted into the cholesterol. Next one, it will be converted into phospholipids. Okay, and also converted into the ketone bodies. Clear? That means very small amount of the fatty acids will be converted into cholesterol, phospholipids, and the ketone bodies. Okay, see these three things. Okay, these are going to be utilized by body. Clear? Totally these three things are going to be utilized by the body. Clear? See, later on these triglycerides will be converted into the lipoproteins. Finally, these triglycerides has to convert into lipoproteins with the help of lipid acceptor protein. Okay, with the help of lipid acceptor protein. Clear? See, until this is going to be happening inside the liver. Okay, all these things are going to be happening inside the liver itself. See, once the lipoproteins are produced, which will be released into the circulation. Clear? As plasma lipoproteins. Okay, it will be released as plasma lipoproteins. Clear? See, that means in the plasma lipoproteins you will call it as LDL and VLDL. Clear? So this is normally, okay, we are discussing the pathogenesis, but here initially you have to learn how the lipids are going to be enters inside the liver, and what are the things that are going to be happening inside the liver, and finally after production of these lipoproteins that is going to be released into the circulation as plasma lipoproteins. See, that is, you will call it as LDL and VLDLs. Clear? So this is generally that is going to be happening inside a liver cell. Okay, but we'll see now what happens exactly in case of a fatty liver. Clear? I think you understood this thing, how the lipoproteins are produced and how it is going to be released into the circulation. Clear? See, if you see lipids, that is as a free fatty acids, which will be, okay, entering inside the liver mainly by two sources. Okay, first one is through the diet as chylomicrons and free fatty acids, and from the adipose tissue. Finally, the fatty acid is going to be enters inside the liver. Clear? See, and not only these fatty acids that is going to be entering inside the liver with the help of these two sources, some amount of acetate that will be present inside the liver also produces this fatty acid. Clear? See, once the fatty acid present in the liver, which will be converted into the triglycerides with the help of alpha glycerophosphate, this also present inside the liver itself. Clear? That means most of the fatty acid is converted into the triglycerides, but small amount of the fatty acids can be converted into cholesterol, phospholipids, and ketone bodies, and these three will be utilized by the body. Clear? Next, whatever the triglycerides will be present, that will be converted into the lipoproteins with the help of lipid acceptor proteins. Now these lipoproteins will be enters inside the circulation as a plasma lipoproteins. Okay, the examples like LDL and VLDL. Okay, this is the normal process that is going to be happening inside the liver cell. And next we'll see what happens in case of the fatty liver. Clear? I told you there is an intracellular accumulation of triglycerides. Clear? In case of the fatty liver, we'll see what are the different reasons. Okay, in case of fatty liver, how there will be accumulation of these triglycerides. Clear? Suppose if there is an increased entry of this fatty acids. Clear? If there is an increased entry of the fatty acids, automatically which will be increasing the levels of triglycerides. Clear? So this is one reason how the triglycerides is going to be increasing. The first one is if there is an increased entry of the free fatty acids, then what happens automatically, which will be increasing the levels of triglycerides. Clear? And next one is suppose increased synthesis of fatty acids inside the liver with the help of acetate. Clear? See, with the help of this acetate, if this fatty acid synthesis is increased, then also increases the triglyceride levels inside the liver. Okay, and next one is decreased conversion of these fatty acids into the ketone bodies. See, if these fatty acids that will be present, if it is not able to convert into these ketone bodies, then what happens automatically, this small amount also there is a chance of converting these fatty acids into the triglycerides. Is it clear? See, that is the third reason. And fourth one is if increasing the levels of alpha glycerophosphate. Clear? If these levels is increased, then what happens automatically, the fatty acids converted into the triglycerides very fastly. And next one is if there is a decreased synthesis of this lipid acceptor proteins, so then what happens, there is no chance of conversion of these triglycerides into the lipoproteins. Clear? So these are all the different factors because of which there is a chance of development of this fatty liver. Clear? See, what is the first one? Increased entry of the fatty acids inside the liver, which will be leading to this formation of these triglycerides, leading to the fatty liver. And next one is increased synthesis with the help of acetate. Okay, first one I told you, because of the entry of the fatty acids is increased, triglyceride levels will be increased. Because of acetate, if it is increasing the synthesis, also the triglyceride synthesis will be increased. And next one is decreased conversion. Okay, that means from fatty acids to ketone bodies is not converted, then what happens, this small amount of fatty acids also there is a chance of conversion into triglycerides. And next one is if this alpha glycerophosphate levels will be increased, then what happens fastly, which will be converting these fatty acids into triglycerides. And next one is if this lipid acceptor proteins is very less, then what happens, whatever the triglycerides that has been produced, which is unable to convert into the lipoproteins. See, finally what happens, these triglyceride levels will be accumulated inside the liver, which will be leading to the fatty liver. Clear? So this is a very simple pathophysiology regarding this fatty liver. Clear? See, if you understand clearly what is happening exactly inside the liver, so that you can easily write the pathogenesis of this fatty liver. Is it clear? I hope you understood this pathogenesis of this fatty liver. Next, we'll see what are the morphological features you are going to be observed in case of this fatty liver. Okay, we'll see the morphological features. See, mainly what happens, the liver is going to be enlarged. First feature is liver is enlarged. Clear? How you know the patient is suffering with the fatty liver? If you see the size of the liver, that is going to be enlarged. Clear? And next one is suppose if the cut surfaces. Okay, see the liver means that it is not always the smooth surface. Clear? There is some cut surfaces. Okay, that cut surfaces are going to be bulges. Okay, cut surfaces are bulges slightly. Okay, next one, this if you see, which is very greasy to touch. See, normally if you see the liver, which is very smooth, clear, the surface is very smooth, but because of this fatty liver, which is very greasy to touch, clear. And next, what happens? The lipid vacuoles in the cytoplasm, clear. See, this lipid vacuoles are going to be developed inside the cytoplasm. Clear? See, once lipid vacuoles means which is the space filled, okay, bubbles. Clear? These lipid vacuoles are going to be present inside the cytoplasm in case of this fatty liver. Clear? See, this lipid vacuoles, okay, initially which is very small in size, okay. Suppose if you see, I'm discuss, okay, will this is one cell, okay, you assume this is one cell, okay, one liver cell, okay, only one liver cell, okay. Here what happens, inside you are having one nucleus. Clear? You're having the nucleus. See, around this nucleus this lipid vacuoles is going to be developed. Clear? Initially, okay, in initially what happens, all these lipid vacuoles will be present around the nucleus, okay. This you will call it as microvesicular, okay. This stage you will call it as microvesicular. Clear? And next what happens in the later stage, see, that means with progression, okay, later on with progression what happens, see, this is also one liver cell. Later on progression what happens is all these lipid vacuoles is going to be pushing this nucleus, clear, to one corner of a cell. Clear? See here what is happening, as these vacuoles are going to be, size is going to be increasing and it is going to be pushing the nucleus towards one corner of the cell. Clear? See, this you will call it as a macrovesicular. This you will call it as a macrovesicular. Clear? So this is regarding the morphological features you are going to be observed in case of the fatty liver. Is it clear? So that means by seeing these stages, you can understand whether it is in initial stages or in the progression stage of the fatty liver. Clear? So in the initial stage means no doubt around the nucleus, the lipid vacuoles will be surrounded in, around the nucleus. But with progression what happens, this lipid vacuole size is going to be increases and it is pushing this nucleus towards one corner of the cell, okay. That you will call it as a macrovesicular. Clear? See, apart from these lipid vacuoles, see, some morphological features like the liver is going to be enlarged and the cut surfaces, okay, you are going to be observe some bulges, okay, slightly you are going to be observe some bulges and the liver is going to be greasy to touch, okay. See, this all regarding the fatty change. Clear? And also we have discussed one example of this fatty liver. Clear? I think you understood regarding what is meant by a fatty liver. Clear? See, later on also we'll discuss in detail regarding the pathogenesis of these alcoholic liver diseases and all those things, but there you remember regarding this fatty liver. Clear? See, that's all students regarding this morphological features, okay. That is morphology of cell injury, okay, regarding the reversible cell injury. Clear? See, until now we have discussed only the morphology of cell injury in case of reversible cell injury. Clear? In the next class we are going to be discussing regarding, okay, the morphology of cell injury, that is regarding the irreversible cell injury.